Mhm. All right. Can somebody confirm you can hear me speaking?
Can hear you.
Thank you. Yep, slap two. That's what I thought. Before we begin, are there any questions? Not hearing anything, so let's begin. Um, I did not finish grading the plagiarism quiz, so I hope to get that done tonight. I don't think I'm finished grading lab one either. And I'll work on that tonight as well. I got a number of responses for the infectious disease project. If I emailed you on it, asking you to pick a topic of the topic you chose, meaning a subtopic, then email me back with your choice of your subtopic. All right, any questions on anything? If not, let me share my screen. I don't think I'm sharing it. There we go. All right, today is October 2nd, and we are supposed to be covering Lab 2 aseptic transfers. Remember, we have begun the infectious disease project, and you need to get me a topic. You need to get your topic approved by me. For one reason, I don't want people to have the same topic. So I'm taking a list of what people have. And then I want to make sure it's an infectious disease. And then I want to divide up the infectious disease if I can divide it up, meaning where you're going to have to pick a subtopic. Like somebody picked, what was it they picked? Somebody picked varicella zoster, and they have to either take chickenpox or shingles. I break that one up into two diseases. There was another disease, I don't remember which it was, where I break it down into early stage disease and late stage disease. And then there was one disease, it wasn't malaria, it was something else, where I actually break it down by the species that causes the disease. I think it was Ebola. And I break that one down by the virus that causes Ebola. There's actually four or five viruses that cause Ebola. The diseases are slightly different. And so I told them you have to choose one of these viruses that cause the disease. You can't write it on all of them. And this way, more than one person can choose Ebola, and then they write it on their specific disease caused by that one virus. Any question about any of that? If not, let's go ahead and begin Lab 2, aseptic transfer. Got that here someplace. There we go. Lab Module 2, aseptic transfers. The learning objectives. One, understand the purpose of using aseptic transfer techniques. Two, determine if aseptic transfer was used from the data provided. Three, define the following terms. What's ubiquitous? What's an aseptic transfer? What is inoculation? And what is a contaminant? Any question about any of that? Let me move this here. In our local environments, microorganisms are ubiquitous, meaning they're essentially everywhere. This is problematic when working in microbiology because we generally want to work with a pure culture, a culture containing one and only one species. Louis Pasteur first developed the technique of getting a pure culture and then using aseptic transfers afterwards to minimize the risk of culture contamination. We'll learn how to make aseptic transfers. Louis Pasteur got an aseptic transfer by making a serial dilution to a, I don't know, contaminated culture, and then taking each dilution and then making 10 replica copies of that dilution. If that dilution had only one tube that had growth in it and 9 tubes that did not have growth in it, Louis Pasteur correctly concluded that the chance of that one out of the 9 tubes being a peer culture was very high, and he assumed it was a peer. And in reality, it was. That was a very difficult way to get a pure culture. And he had to make many different dilutions and then make many replica plates of that dilution and then grow them up and look for one out of the 10 that had growth in it. Edward Koch came up with a much easier way of getting a pure culture, and that is he simply streaked his mixed culture of bacteria onto a solid media, got an isolated colony, took cells from that one isolated colony, and then streaked it out for isolated colonies a second time. If you do it 2 times, then the chance that you will get a pure culture is very high. It's not 100%, but it's greater than 95% chance that you will have a pure culture. Any question about any of that? We actually use Edward Koch's method today for getting a pure culture. All right. During inoculation, some points for maintaining aseptic transfer: never lay the test tube cap or the tube or plate lid down on the surface. I don't know if you can see this. Hopefully you can see my hand is pretending to be a surface. Because if you do, the cap or the lid will definitely pick up microorganisms on the bottom of the plate or the lid. And then when you go and you either take the lid off or you put the lid on, you could have something fall down from the edge of the lid into the plate or into the dish. And then to minimize contamination, you take your tube, and while you're working in it, so let's pretend this is a loop with cells on it where it's yellow. I don't know if you can see the yellow, but and then you put it into the tube. You tilt the tube at an angle and then put the loop in. The reason for putting it at an angle, hopefully you can see this glass here. If you have it straight up, there's a very large surface area that microbes from the air can fall into the tube, and pretending the glass is a tube. If you tilt it at an angle, I have to be careful here because I got liquid in it, which you will have liquid in it. But if you tilt it at an angle, it's a much smaller surface area that you will have microbes that can fall into the tube. It has to come exactly at this angle. If it comes over here, it's going to hit the outside of the tube, which is okay because it's already got microbes on it. Any question about any of that? Another thing you can do to minimize contamination when you're working on a plate, so pretend this is a plate, and you're going to be streaking in the plate, You actually hold the lid over the plate as best as you can, and then move your loop inside the plate during your streak while the lid is protecting the plate. And that way, if a microbe comes down from here, instead of falling in the plate, if you didn't have the lid covering it, it falls on the top of the lid. So it would only contaminate it if it happened to fall right down there. And that's less of a surface area. Any question about any of that? Another thing you do is you hold everything in one hand because you don't want to put that down on the surface because the something can get on the bottom of the lid. So you hold the lid with one finger, usually the pinky, and the hand holding the uh, the loop. And then the other hand, you hold the tube and you hold that tube at an angle so that the microbes coming down, most of them will not fall inside the tube. It would have to fall right here to get inside the tube. And then work efficiently while you're doing this, meaning have the plate uncovered for as little time as possible. Don't talk while you're working with the culture that's open. Obviously, you have to breathe, but best if you don't talk. And then always, before you begin your work, you want to sterilize your environment by disinfecting the area. That will reduce the contamination in the area. And then when you're all done, once again, you remove the contaminants by sterilizing the area. And then you can leave the lab after you remove your gloves, your lab coat, and wash your hands. All right, in order to inoculate, that is, transfer cells into a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. So just cleaning down your work area is a good start, but you still have microbes from the air. And what you do is you have something that'll sterilize something, you have sterile media, and then you use a loop that you sterilize before you put the bacteria on the loop and then inoculate the tube, the sterile tube. And how you sterilize the loop is you put it in the Bunsen burner or the back to incinerator until it becomes red hot. The loop has to be cooled before you can pick up cells. And I think I'll show you how to do that in this lab. What you do is you can let it cool in the air 20 or 30 seconds. That's not the best way to do it because obviously the air has microbes on it. And after it cools, if a microbe touches the loop, it will become contaminated. But if you only cool it for 20 to 30 seconds, the loop is likely to remain sterile. And the best way to cool it is you get it red hot in the flame or in the back to incinerator. And then let's say we're transferring it into this tube of media from this tube that has the cells. You can put the loop inside this tube, touch the side of this tube inside the side of the tube, cooling the loop, and then take the loop to pick up the cells from this tube and then transfer them into that tube. Any question about any of that? And when you're touching the side of the tube, inside, which is sterile, with the wire loop, you quickly cool it. All right, know the terms of this lab. You're not going to be physically performing any of these activities, but you will be watching videos of these activities, and we expect that you will know and understand the material in those videos and in this written lab. So in procedure 1, we're going to transfer sterile liquid using a glass serological pipette. And there's a little video you can watch, watching a proper aseptic transfer technique. I'm not sure if that's the second video or the, oh, that's not a link there. That's the link there. So you're going to transfer 1 ml of sterile water into a tube of nutrient broth, and it'll be 9 mL of nutrient broth, adding 1 ml, and then you'll make a 1:10 dilution. There is a problem when you're transferring water from a pipette or a burette, and that is the water surface doesn't go across the petri dish or the glass burette or the glass petri dish. It might be a plastic petri dish. It doesn't go across flat. There will be a meniscus. That meniscus is particularly large if you're using glass. And that's because the water sticks to the glass and then forms a meniscus. How do you read the water in the pipette tube or in the burette, if it's a burette? Is you read the water not at the top of the meniscus, not for the average or in between the meniscus, but at the bottom of the meniscus. Any question about any of that? And there's the tube of media before we inoculate it with the water. And then we inoculate it with 1 ml of water, and then we incubate the tube for a period of time in the incubator. And then you use the results of this table and the conclusions of this table for the determining what happened for the procedure and then answering the questions below. So if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was successful. If the nutrient broth is turbid, meaning hazy or cloudy, organisms are growing in the broth, and aseptic transfer was unsuccessful. Any questions about that? And then we come down to look at the results, and there's the tube for P1, procedure 1 there. And then you answer the question 0, which I don't think it's shown in your lab module. You have to look at the worksheet. Let me see if I can get to the worksheet here. Come on. There it is, laboratory 2 exercises. So you scroll down. And describe the clarity of the nutrient broth prior to inoculating the tube by adding the sterile water. So what was it? And say if it is turbid or clear, meaning you have to choose clear or turbid. And then for procedure 1, answer question 1. Describe the clarity of the nutrient broth after inoculating with the sterile water and overnight incubation at 35 degrees C. Was the tube clear or turbid? And based on these results, what conclusion can be drawn? Your conclusion must use wording similar to or exactly the same as in the table above. So if the nutrient broth is clear, then the conclusion is no organisms are growing in the broth and aseptic transfer was successful. If the nutrient broth is turbid, hazy, or cloudy, then organisms are growing in the broth and aseptic transfer was unsuccessful. If you use wording much different from the wording on the table, you will get that question at least partially wrong. And you must talk about whether organisms are growing in the broth and whether aseptic transfer was successful or unsuccessful. Any questions about question 1B? Based on the results, what conclusion can be drawn? Then 1C lists 3 sources of error with the aseptic transfer technique that could explain why the nutrient broth showed growth of the microbes. So give me 3 possible reasons. All right. And then there's question 2. Let's go to procedure 2. In procedure 2, you're going to use aseptic technique to transfer a sterile paper disc into a tube of nutrient broth. You have to label the tube and then sterilize your forceps. What you do to sterilize the forceps is you take the forceps. Let me see if I can use something to pretend I have a forceps, because I don't think I have anything exactly like a forceps. Maybe these glasses will be good enough. So we're going to pretend these are forceps because I can move them down. And what you do is we have a container with sterile paper disc. And what you need to do is sterilize the forceps. You can't put these in the flame or the Bacto incinerator until they get red hot, because if you were to do that, you would not be able to hold the metal forceps. Any question about that? So what you do is you put the forceps in alcohol, dip them in alcohol, pretend this is the alcohol, getting the edge of the forceps wet with alcohol, take the forceps out, and then touch the flame, pretend this is the flame or the Bacto incinerator, with the forceps, and that will cause the alcohol to burn off. You then take the forceps that have been cleaned, they're not exactly sterile, but they're clean, remove most of the bacteria, and you go in and you pick up a sterile paper disc, pretend that's a paper disc, and put it into the tube of sterile media. Any question about any of that? And then you incubate that tube containing the sterile paper disc overnight in the incubator. There's the tube before we inoculated it. Scroll down to P2. That's the tube for procedure 2, what the P stands for. After you've inoculated, then you can actually see the paper disc in the bottom of that tube. And then answer the questions. You'll look on the worksheet here. For results for P2, describe the clarity of the nutrient broth after inoculating with the sterile paper disc and overnight incubation at 35 degrees C. Was the tube clear or turbid? So answer the result there. And then based on these results, what conclusion can be drawn? Your conclusion must use the wording similar to the table above. So let's go to the table above. Oops, right here. For procedure 1 and 2, you use this portion of the table. If the nutrient broth is clear, then the conclusion are no organisms are growing in the broth and aseptic transfer was successful. If the nutrient broth is turbid, then organisms are growing in the broth and aseptic transfer was unsuccessful. Any question about any of that? So you answer that. Question 2B right there using the wording similar to the table. And that's it. There is no question 2C here. And then we go to question 3, and it's about procedure 3. Let's come on up here and take a look at procedure 3. Procedure 3, you're going to transfer Serratia marcescens into a broth culture. There's actually a little video for you to watch on this. Oh, there it is there. I was looking for the link for the video. Click on this little link there, and you only need to watch that movie or video until 4 minutes and 23 seconds into the movie. You should note that E. coli is being transferred in the video. E. coli has a different color than Serratia marcescens, but the results should be similar and the procedure is the same. There is an error made in the video when the instructor puts the loop into the E. coli stock tube. So between 3 minutes and 5 seconds to 3 minutes and 20 seconds, you can hear something. You can hear sizzling when he puts the loop into the E. coli. And what was the error that the instructor did? Can anyone figure that one out?
He didn't cool the loop?
Yeah, he didn't cool the loop. So you heard the loop sizzling when he put it into the tube to pick up the E. coli. That could be a problem because if the loop kills all the bacteria that he picks up, he will not transfer any bacteria. In procedure 3, we're using Serratia marcescens. Serratia marcescens is pink when it grows in a broth culture, and it is red when it grows on solid media. So Serratia marcescens has color to it. And then use the results of the table here and then your conclusions. If the nutrient broth is clear in procedure 3, no organisms are growing in the broth. An aseptic transfer was unsuccessful. Why in procedure 3 do we say that aseptic transfer is unsuccessful if the nutrient broth remains clear, whereas in procedure 1 and 2, if the nutrient broth remains clear, we say aseptic transfer was successful? So why are we saying it's different for procedure 3? Can anyone guess?
Is it because we wanted organisms to grow?
Yeah, you want organisms to grow in procedure 3 because you're specifically transferring organisms into the tube in procedure 3. Whereas in procedure 1, you're not transferring organisms, and in theory, you're transferring something sterile into the tube. In procedure 2, you're putting a sterile paper disc. In procedure 1, you're transferring sterile water into the tube. All right. So let's go take a look at procedure 3. Here we see the tube of procedure 3. And you can notice that there's a color to it, and that's from the Serratia marcescens. And then in result 3, describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation. Was the tube clear or turbid? And based on these results, what conclusion, meaning 3b, what conclusion can be drawn? Your conclusion must use wording similar to the table above. And then 3, let's see, C, how do you know whether you transfer the correct microbe, Serratia marcescens, into the tube? There is a way to know. And let me state, if there was only slight contamination in the broth, such that Serratia marcescens was the main bacteria present, but there is slight contamination in the tube, explain why you most likely would not be able to detect the contamination. If there is slight contamination, and the Serratia marcescens is the main bacteria in the tube, it still will contain the pink color from the Serratia marcescens. And you will not see the contaminant, which probably has a different color because most bacteria are not colored pink. There's only a few species which are. And there's, I don't know, thousands of species of bacteria. There might even be tens of thousands or hundreds of thousands. I don't know how many species there are, but there's large numbers. Any questions about procedure 3? All right, then let's move on to procedure 4. Procedure 4, we're going to transfer Serratia marcescens onto a solid media. And in procedure 4, when you transfer Serratia marcescens onto a solid media, it will get the color red. Oh, come on. Okay, why did that come here? Don't know, that's why. There's the solid tube. We're going to transfer onto a slant. When you transfer onto a slant, you should use your loop and then make an S-like movement where you're streaking the bacteria along the slant. This makes it easier to tell if there is contaminant because if it's growing outside where you streaked, like right here, for example, or right there, then you know that's likely to be a contaminant because it's not growing on where you streaked. Another thing that's easy to tell is if a certain region, like this region here, is a color other than red, you know that that colony growth right there, which is not red, is not Serratia marcescens, because Serratia marcescens will grow red. And then based on the results for procedure 3, use this table here. Oh, where did I lose it? Sorry, my mouse jumped. If the slant does not show confluent or colony growth, then no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show confluent or colony growth, then organisms are growing on the slant and aseptic transfer was successful. So here's procedure 4. Even though we streaked this way, you'll note there's so much growth that most of the slant has growth on it. And this looks like there's a liquid here, and that's why this whole region here has growth in it and why it's lighter there. The only region that doesn't have growth is right there and right there, and that's because we didn't streak that region of the tube. We only streaked this region of the tube, and it spread here because the liquid in the tube spread the cells there and there. But here, where it's solid, it didn't spread over to here or there because we didn't streak there. If there had been contaminant, it might be over here or here if it was a different color. So it's much easier to determine if there's contaminants when you're streaking on a slant than if you're streaking in a tube, because you have potentially colonies growing together. And here there's so many colonies here, you don't even see the colonies. They're just, we streaked here and all the area between the streak is growing with growth. All right, then answer the questions to procedure 4. Is there bacterial growth on the slant? You just need to answer yes or no. And does the growth appear, meaning 4B, does the growth appear to be bacterium inoculated or a contaminant? And you need to explain. See if there's another question there. Let's go to the worksheet. So I thought there was another question. Well, that is the worksheet. Nope, there's only 2 questions here then. All right, any question about any of this?
I have a question about the procedure 3
Uh-huh.
questions. On the one that you're showing, it looks like there's 4 questions, the last one being if there was only slight contamination. On the worksheet on Canvas, there's not 4 questions, but
All
there's
right.
questions for
That
the
means I need to update that file. Sorry about that for the fall. I thought I had it updated, but apparently I don't. Let me update the worksheet so that 4th question gets added. So hold off on downloading the worksheet. If you've already downloaded it, you'll need to download it again. Thank you for pointing that out to me. Let me go and doing that right now. In the meantime, you guys can go ahead and get started on well, I guess you can get started on the first part of the worksheet, but realize that that question is going to be slightly changed where there'll be a 4th question. I'll be here until 3:30. Let me go ahead and stop the video. And 2, and then the right one? Maybe. All right, you can refresh your screen and then you should be able to access the worksheet for Lab 2 now. Where is that? Unless I got it to RTF. Let me take a look at that. Maybe that's the problem. Might be going to the wrong file here. Nope, should be going to the right one. Yeah, sorry. All right, I'm going to stop the share, but I'll be here to answer any questions until 3:30 or until the last student logs out. So make sure you ask a question before the last student logs out.